Well, hello and welcome to this lecture. This is going to be on systematics and phylogenetics, and this is our little roadmap. And without further ado, let's get into it. So the first thing we're going to be talking about is eukaryotic cell evolution, since this class is focused primarily on eukaryotes. We've gone through a whole lot of evolutionary theory stuff, and let's talk about specifically of eukaryotes. Eukaryotes started out as some prokaryotic cell, some ancestor, call it a proto-eukaryote. And in order to become a eukaryote, it needed a power source. And we all know the power plant of the cell is what now? Yes, the mitochondria. A little wiggle there, and a little circle there. There's a mitochondria. So, every eukaryotic cell has mitochondria, which are important because they are what produce enough power to keep a large, complex eukaryotic cell going. So, you need mitochondria. However, mitochondria weren't just a natural product of these proto-eukaryotes. The dawn of eukaryotes began with endosymbiosis. Endosymbiosis. So symbiosis is when two organisms have an intertwined relationship. They live within or with each other, right? So eukaryotic cell evolution is a key hallmark of it is endosymbiotic theory. In endosymbiotic theory, this proto-eukaryote absorbed a other prokaryote. So endosymbiotic theory consists of a ancestral proto-eukaryote absorbing a prokaryote. Right? That prokaryote would become the mitochondria. So the prokaryote, instead of getting digested, right? It is not digested. It instead becomes symbiotic. And they enter into a mutualistic relationship. Whoa. Mutualism. In a mutualism type of symbiosis, both organisms benefit. So, the very first instance of endosymbiosis that allowed the evolution of the eukaryotes was absorption of a prokaryote, which then became the mitochondria. So, right here we have a diagram of an endosymbiosis event. Now, it's worth noting that endosymbiosis is not a single event. It didn't happen one and done. Instead, endosymbiosis explains mitochondria, and it also explains plastids, the photosynthetic organelle. So, before I get into all of this, there's specific evidence. Right? So we have the fact that a mitochondria has circular DNA. So our mitochondria, MT there, has circular DNA. That is something only found in prokaryotes. 2, it's approximately the same size as a prokaryote. Right? In addition, it has 2 membranes. A mitochondria has two membranes. One, the inner membrane, is evidence of the original prokaryote membrane. Right? And the second membrane, the outer membrane, is evidence of phagocytosis. Phagocytosis is when the cell's membrane extends and surrounds something and pulls it into the cell and thus has it surrounded by a membrane. So if we have our little prokaryote here, and here's our proto-eukaryote, our ancestor to eukaryotes, and it says, tasty, right? And then it ends up, oh, here's my little prokaryote. Oh, don't kill me, please. And then it ends up fully absorbed. And so you can see now we have two membranes. And so we have circular DNA in mitochondria. That DNA is much more closely related to living prokaryotes than anything in the nucleus. The mitochondria are similar in size to prokaryotes. The mitochondria have the double membrane, which is evidence of that original phagocytosis event. Mitochondria also self-replicate. So during cell division, when you need to crank out enough organelles for both cells, the mitochondria self-replicate. The nucleus is not in control. You feed mitochondria enough, and they'll replicate. So that's pretty cool. As it turns out, photosynthetic organelles also show evidence of endosymbiosis. Now, it's worth noting that all eukaryotes have mitochondria, which indicates that endosymbiosis for mitochondria was the dawn of the eukaryote. But not all eukaryotes have photosynthetic plastids, right? Chloroplasts or the alternatives. So what we have here is an instance where we had an original endosymbiotic event, and then later on in a later lineage, a secondary endosymbiotic event. And that is what we call serial endosymbiosis. Multiple endosymbiosis events over time. Oh my God, I hate this thing. Stop it. So, serial endosymbiosis is where you have multiple endosymbiosis events over time. Multiple endosymbiosis over time. Now, we've got a lot of good evidence that this is how plastids and mitochondria became part of the eukaryote. For one, we've got all that beautiful genetic evidence in which we can sequence DNA. But another great source of evidence comes in protists, which we'll talk about another lecture on. But in protists, we see a large variety of different plastids. So the very first OG plastid, the very first photosynthetic organelle, was likely a protist of some sort combining with cyanobacteria. Cyanobacteria are photosynthetic bacteria. You might know them as blue-green algae. So, the original plastid was likely a combination of a protist and some kind of cyanobacteria. The green plants today don't have that exact combination, but what's really fascinating is when you look in lineages of protists, that plastid ain't the only game in town. So, we have our original, so this is our original primary endosymbiosis in protists. We have our little protist here. And maybe here's our photosynthetic organism. And of course, it grabs it and becomes symbiotic. There we go. There's a specific lineage that has a red primary plastid, and we call those red algae. So red algae have their own plastids separate from the plastids that you're used to. And if we look at a variety of different protists, we see there's a class of protists called cryptophytes. And cryptophytes have what we call a complex plastid, right? The complex plastid. Complex plastid simply means that it is not the product of primary endosymbiosis. Primary endosymbiosis being it's happened once. So, in the cryptophytes here, what we see is evidence that a cryptophyte absorbed a red algae. Now, there's some interesting stuff going on with the cryptophyte plastid. For one, there's the outer membrane of the cryptophyte plastid. Then there's an inner membrane. Then there's this little region of reduced linear DNA. And then there's another membrane. And then there's another membrane. Right? So we have 4 membrane layers. And what we see here is this reduced linear DNA that we talked about over here is evidence of the red algae's nuclear DNA. So what we have is the original plastid of the red algae right here, and then we have what's left of the cytoplasm of the red algae and what's left of its nuclear DNA right here. And then this outermost membrane is the phagocytosis membrane, so the phagosome membrane. So that's secondary endosymbiosis, right? Where we have a organism absorbing an organism that had already undergone endosymbiosis and undergoing endosymbiosis. And what's really fascinating is that when it comes to a group of organisms called the ochrophytes, their complex plastid indicates that their ancestor absorbed one of these cryptophytes to create their plastid. And then what's really fascinating is that these haptophytes have tertiary endosymbiosis. So that's really cool. We've even found evidence of quaternary endosymbiosis, which is pretty amazing. Right? In tertiary endosymbiosis, we have 6 membranes around the plastid, hypothetically. In quaternary, you could have up to 8 membranes surrounding the plastid. So these protists have all this beautiful genetic evidence and molecular evidence of multiple different endosymbiosis events occurring throughout different lineages of protists. So we definitely have plenty of evidence for this to be a scientific theory. Extremely well supported by multiple lines of evidence. All right, so now that we're in eukaryotes, let's classify them. So early on, I might have mentioned the 3 domains of life. If I haven't already, we separate all life into 3 basic domains. Domain Eukarya, domain Archaea, and domain Bacteria. So bacteria are pretty easy to recognize, right? Anything that's a bacteria belongs in domain Bacteria. Oh my God, brain is breaking. Archaeans are also prokaryotes, like bacteria. They are generally prokaryotes that can survive in extreme environments. So we often call these the extremophiles. And then lastly, we have eukaryotic organisms, and they are all grouped into domain Eukarya. So within domain Eukarya, we then separate all life into 6 distinct kingdoms. So we have fungus, plantae, animalia, and then protista. This is... I'm just going to cut this in post. So we have those 3 domains. Within those 3 domains are 6 kingdoms. 2 being accounted for by the domains Archaea and Bacteria. Eubacteria, kingdom Eubacteria in Bacteria. EU basically is a prefix we add when we want to say like true. And then kingdom Archaebacteria for domain Archaea. So the 4 remaining kingdoms are Protista, Fungi, Plantae, Animalia. Now, past that, we separate it into continuously smaller categories. So as we move along in classification, we will create organized categories. So a taxa is the plural of taxon, and a taxon is basically just all of the members of a group at a certain level of classification. All members at a specific level. So, for instance, the taxon Eukarya includes Protist, Fungi, Plantae, and Animalia. The taxon Animalia includes all the animals, right? So, taxonomy itself is the discipline in which organisms are named and classified based on relationships. Usually evolutionary relationships. In the past, relationships assuming relatedness based on similarity. So we start off with kingdom, right? Start off with domain, then we have kingdom. But the rest of it is really only useful in Eukarya because Bacteria and Archaea get real fuzzy when you try and go into these levels. So after kingdom comes phylum. So here's phylum. There are multiple phyla of animals. After phyla comes class. After class comes order, then family, genus, and then the most specific classification, species. So, phylum, class, order, family, genus, species. So, kingdom, king, phylum, Philip, class, came, order, over, family, for, genus, great, species, spaghetti. King Philip came over for great spaghetti. Oh, what mnemonic up in the house? All right, so modern taxonomy is naming species based on something we call binomial nomenclature. In binomial nomenclature, what we are saying is that any given species has two basically names, right? So a species name is based on both the name of the genus that they belong to and then the name of their specific species. For instance, humans are Homo, we are genus Homo, species Sapiens. Now, you always uppercase the genus name and lowercase the species name. Genus is always capitalized, species is never capitalized. This is from good old Carl Linnaeus. So Linnaeus's original OG classification system is the one that we pretty much still use. And so he was the one who came up with the binomial nomenclature to identify specific species. These are generally Latin or Greek-derived words, so you get to do things like pronounce it with confidence. So the giant anteater is Myrmecophaga tridactyla. The Chuckwalla fat dark lizard is Sauromalus obesus. Over here we have the African hedgehog, Atelyryx albiventris, ineffective fighter with a white belly. So that's pretty amusing. Although it is worth noting that hedgehogs have an enzyme running in their body that can neutralize the venom of vipers. And adult hedgehogs will often hunt down and eat young vipers. And if they get bit by the viper and get a whole bunch of venom injected into them, they don't really care. They'll slow down for a bit while the enzymes in their body work on it, but eventually the venom is neutralized. So I don't know if they're really all that ineffective of a fighter. Aceros plicatus, sharp point with wrinkles, is the Papuan hornbill. Look, wrinkles. Anyway, so this is how we characterize species. All right, now when we want to arrange organisms together in a taxonomic tree, we call this tree of relationships a phylogenetic tree. It's a diagram that represents the evolutionary history of a group of organisms. The original trees were based on physical similarities. Now we can create different kinds of trees, including ones based on genetic evidence. So, systematics is specifically the reconstruction, right? Reconstruction and study of evolutionary relationships. That is what systematics specifically is. So a phylogeny is essentially the evolutionary history of a species or group of species. Of one or more related species. So when you create a phylogeny, you are trying to determine the evolutionary relationships between organisms. Right? You can have a phylogeny depicting the entire animal kingdom, a phylogeny trying to depict all 3 domains and all the organisms within it. You could have a phylogeny specifically of primates, right? So here is our picture of a primate phylogeny. Specifically, when you take a phylogeny and put it in a diagram form, that's when you have a phylogenetic tree. So the word phylogeny on its own doesn't imply that you're making one of these pretty trees. A phylogenetic tree is a diagram of a phylogeny. So phylogenies in general are hypotheses about those evolutionary relationships. A hypothesis about the evolutionary relationships. Once you create a phylogeny, you must then collect evidence to support your phylogeny. So it uses various features or characters to construct these relationships, right? These can be physical appearance, which whether it's external or internal, we'd call this morphological evidence, right? So it can be based on the morphology of the organism. It can also be chromosomal evidence, right? So using DNA, either chromosomal DNA or mitochondrial DNA, so genetic evidence. And you can even use various kinds of other molecular evidence. So you might compare proteins instead of outright comparing DNA. So the tree that you build out of it, the phylogenetic tree, is a great depiction of descent with modification, right? Where you show common ancestry, right? So each point on the tree, each branch is representing divergence from a common ancestor. So right here we have a common ancestor, and we have one lineage and another lineage. And then here's another common ancestor with 2 lineages diverging. So we assume that the common ancestor to gibbons, orangutans, gorillas, humans, and chimps is pretty far back, and then based on similarities, we depict how the lineages descended from one another. And we can have various different trees that we can build on it based on your hypotheses. So it's a great way to depict common ancestry. This picture on the left over here is actually an image of the sketch Darwin made in his notebook for this very concept, which is pretty cool. So how to read a phylogenetic tree. We've got some very distinct vocab to go on here. One, we have little points that represent common ancestors. These points are called nodes. So the nodes are common ancestors, right? From each node, you assume there's a branch point where lineages divide, right? So from the common ancestor, we have a divergence of lineages. Sometimes it's pretty easy. You have a single node and 2 lineages coming off of that node. We would refer to these 2 closely related lineages sharing one single common ancestor as sister taxa, right? So sister taxa two lineages sharing a single recent common ancestor. So you only have to go back to one common ancestor. All right, so in other cases, you have a node and 3 branches coming from it, right here. So when you have more than 2 branches coming off of it, it is what we call a polytomy. So 3 or more branches from a single common ancestor. So 3 or more branches from a single common ancestor. So these are still taxa that share one single recent common ancestor. They're all 3 have one single common ancestor. But because there's 3 or more, we call it a polytomy. Right? So we've got nodes, branch points, sister taxa, and then what we want to talk about are the start of any tree, the basal taxon. So this is a lineage that diverts very early. So you always have some kind of ancestral common ancestor at the start. That's the common ancestor to everything in your tree. And then if you have a single lineage that branches off very early from it, we would call this lineage a basal taxon. So it's going to be representative of a much more primitive member of that group in that phylogenetic tree. So this is how we read a phylogenetic tree for those evolutionary relationships, using nodes, branch points, identifying sister taxa, identifying polytomies, and identifying basal taxa. So now that we know how to read a phylogenetic tree and where common ancestors are and sister taxa and things like that, we can look at some of the interesting variations of how you group organisms in a phylogenetic tree. So when you have a phylogenetic tree, you can group things differently. So one, you can look at a group of organisms with a single common ancestor, right, in which all organisms that you're examining have a single common ancestor. We would call that monophyletic. All in the group share a single common ancestor. You could also look at a paraphyletic grouping, right? So in a paraphyletic grouping, you can see that most of the organisms here all stem from one common ancestor. So we've got this one common ancestor, and we're examining all these organisms stemming from it, but we aren't examining all of them, right? So our grouping ends here. And our group does not include lineage A and lineage B, which are also part of this original common ancestor in our group. So in a paraphyletic grouping, you've got a group, a taxon, in which we have a single common ancestor, but not all descendants. So some, but not all descendants from that common ancestor. And then lastly, we can have a polyphyletic grouping. In a polyphyletic grouping, we do not have a single common ancestor to represent our group. Right? We are looking at groups that do not have a single common ancestor. So because here you are looking, where's my pin? Come back here. Okay, so here you are examining E, F, and G. And these all share one common ancestor here. And then in this group, you are looking at C and D specifically. And they share a single common ancestor here. So because you are examining specifically these two different groups in your taxon, you do not have a single common ancestor. You'd have to come back all the way to the bottom to get a paraphyletic grouping of all of these. So because you are examining just one common ancestor back on them, you have a polyphyletic grouping, because each one of these you're going one back, so that you have two different common ancestors. Right? You do not have a single shared common ancestor. So let's look at some pictures. This is a grouping of the archosaurs. So the archosaurs are a group of organisms that include the dinosaurs and, crocodiles. So the archosaurs are dinosaurs, both avian and non-avian. So here's all our dinosaurs. And the crocodilians. That's the archosaurs. You can see we've got this large grouping, and it comes back to a single common ancestor. Every single member of the group you have can trace back to a single common ancestor. So this is a monophyletic group. Then you might look at the non-avian dinosaurs. Non-avian, thank you very much, dinosaurs. In which case you have a paraphyletic group, because here's a common ancestor, right? And you have three lineages of non-avian dinosaurs, but you are leaving out the avian dinosaurs. So you have some, but not all, descendants from a single common ancestor. That is paraphyletic. Then here is where you're looking at a group of flying vertebrates. So these are avian dinosaurs and flying mammals. So the flying mammals obviously have their lineage with mammals. So you've got one group here with its common ancestor. And then you have the avian dinosaurs, and their lineage is down there with the rest of their dinosaur friends. And this is two separate groups, and you have two separate common ancestors. You do not have a single common ancestor. So this is a polyphyletic grouping. And now you can use phylogenies to build hypotheses about evolutionary history. right? So you can use a phylogeny to build a hypothesis about evolutionary history. So our example here is larval dispersal, the evolution of larval dispersal in marine snails. This is our organisms, and we can see that there are two basic ways you can have larvae in marine snails. You can have larvae that are dispersed in currents that become part of the plankton in the ocean, and you can have larvae that are non-dispersed, larvae that move down to the substrate and hang out in a single area. They are not carried in the currents. So we have dispersal, we have dispersed larvae and non-dispersed larvae. So marine snails have either dispersed larvae or non-dispersed larvae. Looking at the fossil record, we can see the percentage of non-dispersing larvae, right? So in orange here, we have our percent of non-dispersing larvae, orangey red, whatever, ochre. And in the fossil record, we see very few of them throughout most of the Paleocene, right? A slight increase. But then when we get to the Eocene, we get a big increase in those non-dispersing larvae. But we can build some hypothetical phylogenetic trees to try and develop a prediction for how this happens, right? Fossils show an increase in non-dispersing snails. So the question is, why is there an increase in non-dispersing snails? And we can have two basic hypotheses about this. One, non-dispersing snails have greater evolutionary success, and they survive more extinctions. So that could be one reason why we see more non-dispersing snails. They have greater evolutionary success and survive more extinctions. An equally valid hypothesis might be that they evolve, they tend to evolve from dispersing snails and dispersing snails do not tend to or evolve from non-dispersing snails. So, in other words, a non-dispersing snail will tend to evolve from a dispersing snail. If you have your little evolutionary relationship and this is your dispersing snail, you might get a non-dispersing snail evolve from it. And however, you tend not to see the reverse in this hypothesis, where a non-dispersing snail produces a evolutionary lineage that would result in a dispersing snail. Right, so you tend not to see the reverse. This one, the first one, seems like it might make a little bit more sense. They're evolving more often, they're having greater success, they're surviving extinctions, and that's why you see them increase in number. But the reason this is an equally valid hypothesis, just that dispersing snails evolve from non-dispersing snails and not generally the reverse is because you have a chart of 25 million years from 65 to 40 million years ago, and assuming that these snails, these marine snails in general, are successful, a byproduct of non-dispersing snails evolving from dispersing snails will just necessarily be an increase in non-dispersing snails over time. You've got 25 million years to evolve non-dispersers from dispersers, so you're going to get more non-dispersers. And because you don't tend to go from non-disperser to disperser, that number will just sort of generally increase. So that is why these are two equally valid hypotheses. One is that they have greater success and they survive extinctions much more effectively. And the other is that simply non-dispersing snails evolve from dispersing snails and you don't get the reverse. So you have two basic hypotheses for those phylogenetic trees, right? Here in hypothesis A, we have an evolutionary change from dispersing to non-dispersing occurs in one direction. We go from dispersing to non-dispersing, and rarely in the opposite. So just as a byproduct, you could have, your common ancestor here, and then you have these lineage diversions, and then you'll have a lineage of non-dispersing, and some more, and some more, and more, and then you have non-dispersing, and non-dispersing, and non-dispersing, right? So you just get, over evolutionary time, new non-dispersing lineages. So then for B over here, we have that non-dispersing snails evolve more frequently and become less frequently extinct. So they more frequently evolve, become less frequently extinct. So if that were the case, what you'd see, what you'd expect is significantly greater amounts in modern species of these non-dispersing snails. So here, in hypothesis A, where you're just saying you see more of them because you've got a lot of time for them to evolve, and then it doesn't tend to go backwards, you might not necessarily see more non-dispersing than dispersing. You just see an increase. Over here in B, because they are evolving more frequently and having greater success, you would see eventually more non-dispersing than dispersing. So in this case, we've got 1, 2, 3, 4, 5 lineages in this picture of non-dispersals. Then 1, 2, 3, 4, 5. With that, so there's 6, and then all these lineages surviving to modern times. Right, so in hypothesis A, you don't necessarily have to have more non-dispersers than dispersers. You just have them evolving and increasing in frequency because of evolutionary time. Whereas here, you would see not only an increase in frequency of non-dispersers, but a decrease in frequency in dispersers. So when you actually develop a phylogenetic tree in which you look at all the organisms, dispersing and non-dispersing, what you see is that there are a significant amount of marine snails in blue that disperse their larvae. So you can't really say that they have a competitive disadvantage. because there are just so many species. So the fact that we also see these non-dispersal lineages occur, and we don't really see any branches coming from them that produce dispersing lineages, it would seem that the proliferation of non-dispersing larvae appears to just be a consequence of evolutionary time and the fact that dispersing snails with dispersing larvae tend to produce snails with non-dispersing larvae, evolutionarily. Okay. Now, knowing evolutionary relationships, in other words, how closely things are related to each other evolutionarily, can help us develop hypotheses about other things, such as behavior and other physiological traits. So, for instance, if we're looking at these phylogenetic trees of evolutionary relationships, and we look at the relationships of reptiles, we're not including turtles and tortoises, so this is going to be paraphyletic, but we have our lizards and snakes here, and then we have our crocodilians, then our ornithischian and saurischian non-avian dinosaurs, and then our avian dinosaurs. Now, because both avian dinosaurs and non-avian dinosaurs are so closely related, and because crocodiles are closely related, we might be able to make some basic assumptions about behavior. What do, for instance, reproductive behavior-wise, what do these four share in common? Nesting behavior. Right? So they all build nests. Even lizards build nests. But another thing they all share would be brooding. Lizards and snakes are less likely to show brooding, where they build a nest and then sit on it, right? Either to protect it or warm it. Right? The closest relationship is between birds, the avian dinosaurs, and the non-avian dinosaurs. So if we wanted to make some hypotheses regarding dinosaurs, we could make some. Right? So perhaps the non-avian dinosaurs had 4 chambered hearts. All avian dinosaurs have 4 chambered hearts. So it doesn't seem out of the way, all that crazy to think that the non-avian dinosaurs would have 4 chambered hearts. How about vocalizations? All the avian dinosaurs have different levels of vocalizations. So perhaps dinosaurs, the non-avian dinosaurs, vocalized. If they have interesting little adaptations on their head, maybe they could even have songs of some sort, like a bird song. Nesting and brooding. Right? We could make all these as valid hypotheses simply based on a tree depicting their evolutionary relationships. And then you could examine the fossil record to find any evidence for your predictions. For instance, over here we have a fossil of an oviraptor with eggs. So this oviraptor, based on the body positioning, right, the bones are surrounding the eggs, right? So it appears that the dinosaur may have died while brooding or protecting the eggs. Birds warm their nests. Crocodiles guard their nests and warm their nests. So, evidence like this supports the idea based on both evolutionary relationship and the fossil record, support the idea that at least some of the non-avian dinosaurs exhibited nesting and brooding behaviors. So, and because those are there, you're more likely to also support the hypotheses that these non-avian dinosaurs exhibited four-chambered hearts and perhaps vocalizations of some sort. All right, so cladistics. In cladistics, we are creating a phylogenetic tree and we are including on it important characteristics. So in our phylogenetic tree, we have important characteristics. Now you might recall when we were talking about adaptive radiation, when one species showed increased speciation to fill multiple roles, we talked about key innovations. So whether it's behavioral, morphological, genetic, whatever, a key innovation is a feature. So in this case, in cladistics, we'll often use a key innovation, specifically an identifying trait that we can use to identify that lineage. So when you build a cladogram, which is a phylogenetic tree with characteristics on it, you are going to have shared derived characters. Shared derived characters are basically a character where all lineages beyond that character share it. So, that characteristic was derived from some ancestor before, and all the lineages share that characteristic. It's a shared derived characteristic. So, this is based on similarities that are inherited from the most recent common ancestor. Inherited from the most recent common ancestor. So when you have a cladogram like this one here, when I put down vertebra and jaws, right? That means that it is a characteristic derived from whatever this common ancestor was. And it is shared amongst all lineages beyond. And then the next character will describe, will be a key innovation for the, a key identifying innovation for the next division. So, vertebra and jaws includes fish, like the bass. So if you want to then separate out your land animals, you might need 4 legs and eggs. Right, so this is our common ancestor that has all of these vertebrae and jaws. Then we have 4 legs and eggs, and here's the common ancestor and all the organisms derived from it that share that characteristic: lizards, horses, and monkeys. So then if you want to go into mammals, you have hair and mammary glands as our characteristic that's derived from whatever this ancestor to lizards and mammals was. And there we go. There's the common ancestor and all the lineages that have it in our cladogram. So, these are all monophyletic. A cladogram is always, to a T, monophyletic. Cladogram is always monophyletic. So we always start a cladogram with an outgroup. So an outgroup is a group that is closely related but does not have any of the shared derived characteristics. So in our example here, this is our outgroup. We haven't listed any characters for our outgroup. So this is Amphioxus. And it is our outgroup. All right. So we always put that on there to create sort of a more specific cladogram so you can get a better idea of the evolutionary relationships we're looking at. This here is the Amphioxus. It's a chordate, which means it has some similar features like to these organisms, sort of a recent ancestor. So some of the things that Amphioxus has that all of them in the cladogram have would be a dorsal nerve cord. Really? Gonna erase my letters? Really? A dorsal nerve cord. Right? Gills. A post-anal tail. And a oh my goodness. Ugh. Notochord. Just ran out of space up there. Notochord. We'll talk about the notochord later. We can just sort of ignore that. But the point is that both the outgroup and everything else have all four of these. So none of these four characteristics are useful in separating out the lineages in this cladogram. So these are called ancestral traits, shared by both the outgroup and everything else. So these are traits that arose prior to the common ancestor in your phylogeny or cladogram. So in this case, because your cladogram is for fish like the bass, lizards, horses, and monkeys, right? Your common ancestor is basically right here. And the ancestral traits would go here before the outgroup. So we're not really using ancestral traits, we just identify them. So the plesiomorphies are traits of the ancestral state. You don't need to include plesiomorphies in your cladogram, right? Because everything in your cladogram has that, including the outgroup. You don't need to include that. So primarily, what you put down are those derived traits. So a derived character is something derived from that ancestor that gave rise to this new division in your lineage. So the term for a derived trait is a synapomorphy. So a plesiomorphy is an ancestral trait. A synapomorphy is a derived trait. A group of organisms that share a derived trait, that share a synapomorphy, are called a clade. So A clade, I'm running out of space here. A clade shares synapomorphies. So for instance, in this cladogram, we have bass, lizards, horse, and monkeys. This is the clade of vertebrates. They have vertebra and jaws. Those are the vertebrates. Then we have lizards, horse, and monkeys. This is a new clade. This clade shares forelegs and amniotic eggs. These are the terrestrial vertebrates. And then lastly, the horse and monkey, which share hair and mammary glands. This is the clade of mammals. So a cladogram will have multiple clades because you tend to have multiple synapomorphies in order to separate out your lineages. So here we can see in our we have the classic phylogenetic tree style drawing. And it's especially well pictured with these boxes that you have a nested hierarchy where basically you have traits, the identifying features create smaller and smaller categories within your cladogram so that each one is nested in the other. So your horse and monkey share hair and mammary glands, but they're nested in with lizards because they also have forelegs and amniotic eggs. And lizards, horses, and monkeys are nested in with bass because they all have vertebra and jaws. So we have a nested hierarchy. So clades within clades, where we go from vertebrates to terrestrial vertebrates to mammals. It's like those nesting Russian dolls of dividing organisms. Whew. Okay, so in cladistics, when you're building a cladogram, it's important to identify what organisms share what traits, right? Only the shared derived characters are considered informative, right? These are the only ones that are considered informative about the evolutionary relationships. This is because an ancestral trait is shared by the entire tree. So why would an ancestral trait give you any information about the evolutionary relationships within the entire clade you've got? So this is another clade similar to the one we had. The lancelet is our primitive outgroup. So we don't need to have postanal tail, dorsal nerve cord, gills. Everything else has that. So the outgroup and everything past has that. So we don't need that listed as a shared derived character. Those are the ancestral traits. Whoa. Come back here. So only the shared derived traits are considered informative, and that's why those are the only things we put on the phylogenetic tree. These traits must distinguish your groups from their ancestors. So when you build your cladogram, your shared derived traits are distinguishing traits from the ancestral state. So a vertebral column, the presence of a vertebral column, distinguishes all of the organisms in our tree from the outgroup. So it is a distinguishing characteristic. So we're always doing derived traits versus the ancestral traits. And so, of course, branch points beyond a derived characteristic share all that. All branch points past a shared character share that character. Like, that's, I'm not even sure why I'd write that down, but I have that there. I get repetitive sometimes. So, all branches past the synapomorphy share that synapomorphy. So you have to position your synapomorphies on the tree properly. So oftentimes what people do is they make a little chart, right? You'll do that in lab when you make a cladogram. So here's a little chart. You build a table, and you put your synapomorphies in the table: vertebral column, hinged jaws, 4 walking legs, amnion for eggs, and hair. And then you just mark down what shares those. So if you set it up correctly, your outgroup will have none of your synapomorphies. It shouldn't have any of the synapomorphies. The outgroup is representative of the ancestrals. So then you just put how many have them. So your first synapomorphy should pretty much distinguish everything from the outgroup. And then from there on, you get more and more detailed, right? Now, there are cases where you'll have a characteristic that is shared by only one of them in there. So, for instance, with amphibians, they undergo metamorphosis. So, because none of the others have a larval tadpole stage that then undergoes metamorphosis to a terrestrial stage, what you'd do if you wanted to include that is just, probably here's a better spot. Just put that branch there. So, aquatic to terrestrial metamorphosis. And there you go. So, there's a synapomorphy specific to the frog. And so you'd put it here, and that way, like, if you had salamanders on there, boom. So, you'll encounter that in lab. All right, so looking at a cladogram, does the length of the branches matter? So, the answer to that is it depends if you want them to matter. Right? A perfectly valid answer could be no. For instance, all these cladograms we've been developing over here are just built to show the synapomorphies and everything coming off the synapomorphies. Right? So there's your hair. And so this branch length doesn't really tell you anything beyond that we wanted everything to be right justified here. And so all the branches end up on the right side. And it doesn't really tell you anything. Right? So, no is a perfectly reasonable answer. Branches can mean nothing. Branch lengths can have no meaning. Now, branch lengths could also represent genetic change. So, amount of genetic change. So right here is a cladogram indicating some genetic changes on there. So the longer the branch, the more genetic change. So in this case, you can see that everything's not perfectly right justified. The branches are of varying lengths depending on the amount of genetic change from the most recent common ancestor. So here's your outgroup Drosophila, right, which is an invertebrate. So it's got a fair amount of genetic change from this common ancestor to the next one. And then you have the start of your chordates. So then you have this common ancestor, and there's actually quite a bit of genetic change between the lancelet and, the zebrafish. However, we can see that there's less genetic change between lancelets and zebrafish than between zebrafish and Drosophila, because the branches go back pretty far, right? You have to go all the way over to here to get that genetic change. So the shorter the branches between ancestors, the less genetic change between them. So comparatively speaking, humans and mice have very little genetic change in this particular scope, because it's all chordates. So they're both mammals. So when comparing the amount of genetic change between human and mice and human and chickens, there's very little. The other thing you can illustrate with your phylogenetic tree is time. How far back in geologic time do you have to go before you get to the most recent common ancestor? Most recent common ancestor. Well, I get better with tablets over time. So here we can see we have our eras in millions of years ago. So here's the Cenozoic from 65.5 to the present. The Mesozoic is from 251 million years ago to 65.5 million years ago. The Paleozoic is from 542 million years ago to 251 million years. So to look at the amount of time that has passed since the most recent common ancestor between humans and Drosophila, we have to go all the way back to the dawn of the Paleozoic, whereas humans and mice only diverged near the beginning of the Cenozoic, whereas humans and avian dinosaurs diverged out there way back still in the Paleozoic. So you can get those time to most recent common ancestor with branches. So branch lengths can mean nothing really. They can also represent the amount of genetic change between the organisms in your cladogram, and they can also be put on an axis with time and represent time to most recent common ancestor. Getting on there, it's this slide and two others. All right, parsimony and likelihood of relationships in your cladogram. All right, so parsimony. You might know parsimony from the sort of common phrasing term. People like to throw out Occam's razor. The simplest answer is is usually the correct answer. That's not really the best way to explain it. So parsimony is the principle of finding the answer with the fewest assumptions. So the principle of maximum parsimony in cladistics is the principal idea that you want a cladogram or a phylogenetic tree with the fewest common ancestors. And that is the most likely representation of your relationships. So if you look at a table here with the percentage of differences between gene sequences, comparing humans, mushrooms, and tulips, in other words, animals, fungus, and plants, we can see that humans have about 30% different genes from fungus, and humans have about 40% different genes from plants. Fungus have about 40% different genes from plants as well. So we could build two potential cladograms from this, two potential phylogenetic trees, illustrating the percent of genetic differences between entries on the tree. So we'll start with the one on the right. Here's our common ancestor between fungus, humans, and plants. And in this case, we have a branch representing a 5% genetic difference from fungus to this common ancestor. And then a 10% difference from that common ancestor to the common ancestor between humans and plants. And then another 15% difference to humans here. So this gives us 5%, 10%, and 15% adds up to the 30% genetic difference between humans and mushrooms. And then to get from humans to plants, right, we need a 15% difference here and a 25% difference here to get us to the 40% difference. And that should also work for our fungus. So 5, 10, 25, that's 40%. Right? However, you could also build a phylogenetic tree where we separate these out. Now, because the genetic differences between humans and mushrooms is only 30%, we might assume that humans and mushrooms are more closely related to each other. So here we go. We have a common ancestor and a 15% difference between humans and mushrooms. All right, so that from here to here gets us our 30% difference. Then you have a 5% difference from this lineage to the common ancestor that produced there, so that gets us 15, and 5 is 20%, so then you just need that 20% difference to plants. And this occupies our principle of maximum parsimony. We have the fewest genetic changes required to get our different lineages with their different relationships. So, this would be the more likely phylogenetic tree because it has less assumptions in how those common ancestors diverged. In our less likely tree, we have to assume that there's a common ancestor that only required a 5% divergence to get to fungus, and then a 10% to a common ancestor between humans and plants, which then has a major genetic difference between humans and plants. That seems incredibly unlikely. Because there's only a 30% difference between humans and mushrooms, it's far more likely that it's humans and mushrooms that share the most recent common ancestor in this group. And then you have to go a level back to get to the common ancestor with plants. So you're trying to build a cladogram or a phylogenetic tree with the fewest common ancestors, the fewest assumptions required to get that tree. And that is your most likely tree. So, you probably all know this as Occam's razor. However, most people likely don't always say it correctly. Like, a lot of people like to bust out Occam's razor and say that the simplest solution is the most correct one, and it's that the simplest solution is most likely the correct one. Gotta make room for probability being a jerk. Almost done, people. Almost done. Okay, so let's get into some of the problems we encounter with building cladograms. So when doing cladograms, monophyletic groupings do not always make sense, right? Monophyletic groupings don't always make sense. For instance, with hominids, right, we have primates that are bipedal, grasping hands. Bipedal means they walk on two legs. They have grasping hands. You generally have large brains, right? So bipedal, large brains, walking on, yeah, so that's hominidae. And we separate hominidae from pongidae, which we refer to as the apes, right? The great apes have no tails, right? They tend to have a quadrupedal stance in which they have shorter back legs and do a lot of knuckle walking, right? That's Pongidae. And then out here we have Hylobatidae with some of the monkeys, right? And so monkeys are arboreal. They live up in trees, swing around. They tend to have very long arms and long legs. They have tails, right? So this is a nice little cladogram, a phylogenetic grouping of animals based on characteristics that make sense, based on unique adaptive zones, right? So anyway, problem here is that when you look at genetic relatedness, orangutans belonging to Pongidae are more closely related to Hylobatids than they are to chimps. And chimps are more closely related to humans than they are to gorillas. So we have a grouping based on shared characteristics that certainly makes sense when you look at a lot of these shared characteristics. But at the genetic level, this grouping doesn't make a ton of sense. Why put orangutans in the same family with gorillas and chimps, if the orangutans are much more closely related to monkeys than they are to chimps? Why would they be in the same family as chimps? Why would humans be out on their own in hominidae if they're much more closely related to chimps, right? So these are problems with this sort of thing. You have to choose your characteristics, and it doesn't always make a perfect phylogeny, right? So another issue comes to homology and analogy. right? So when you are building your phylogenetic tree based on shared characteristics, shared derived characteristics, these are often external appearances. And you can run into a problem trying to build a phylogenetic tree based on evolutionary relationship, based on shared characteristics, because of convergent evolution. Right? This is a marsupial mole. This is a placental mole. If you're trying to build a phylogenetic tree based on key characteristics such as nearly blind, large nose, highly sensitive like highly sensitive whiskers, large claws for burrowing, and you have like, so say you have mice and rabbits and moles and marsupial moles, you would be tempted to put moles and marsupial moles very close together. So, because they have very similar characteristics. But these characteristics were developed via convergent evolution, right? Your phylogenetic tree can't look like this. It would have to be much more like this. All right? So, when you're looking at external characteristics, convergent evolution can present a problem to developing correct phylogenetic relationships. In fact, Carl Linnaeus's original classification trees are getting reordered with modern genetic techniques. We are having to pull things out of families that they used to be in and put them in different families based on genetic relatedness, because a lot of our taxonomy, a lot of our phylogenies are based on external characteristics. And homology is where we have shared similarities due to shared ancestry. From shared ancestry. And analogies are similarities with no shared ancestry. Well, no recent shared ancestry. Analogy is also called homoplasy. So analogies and homoplasies are basically synonyms. So similarities without shared ancestry, recent ancestry, and similarities from shared ancestry. So if you're looking at this, all these shared limbs, like all these shared bones in the limbs of these vertebrates, are homologous. But if you're a naturalist from days back, they wouldn't appear all that similar. They have a radically different appearance. Human arms, bat wings, horse legs, and whale flippers, right? They don't appear all that similar. So you wouldn't necessarily know that they're homologous based on the external appearance. They share similar characteristics even though they are phenotypically different. That's their homology. Whereas over here, we share phenotypical character. We share a similar phenotype even though we do not have recent ancestry. So, when building a phylogeny, especially in the days of old, homologies and analogies and convergent evolution, producing those analogies, could present a problem. You could produce incorrect cladistics because you misinterpreted analogous bodies and homologous bodies. So that's it. That's systematics and phylogenetics and cladistics. So you'll be doing a worksheet that involves this stuff. So now you know everything you need to know.